Hello and welcome to the final presentation. Nope. Hello and welcome to the presentation of the final project for deep learning for healthcare. This is the presentation. Hello and welcome to the presentation. Hello and welcome. Hello and welcome to the presentation. Hello and welcome to the presentation for 3J1M. Hello and welcome to the presentation by 3J1M, also known as Team 1503, for deep learning for healthcare. We chose drug discovery as our primary topic and worked through recreating several methods. Drug discovery is a broad topic. It's a topic that started often just finding ways to try and reduce the search space that chemists and pharmaceutical companies really need to search to find things. There's one topic that remains valid really throughout all of our methods, and that's how do we go about representing a molecule? There are a couple different ways of doing that, and the methods have changed over time. But all of these methods will see a turn towards using some sort of latent representation in variational autoencoders, graphs, as opposed to representing them in a more naive way like a raw smile string, which we'll get into in a bit. But deep learning methods allow several techniques which may not have been possible or feasible. We see that with some of the advancements with AlphaFold and the like. Being able to predict how well a protein's affinity for a particular molecule and what that can mean for treatment methods. Ways of using existing drugs and modifying them in particular ways, trying to find candidates that do the same thing but do something else. And that's really where we focused our methods, is on number 4 in the diagram on the left, which is de novo design. What are methods that we can take, what we know, and find new molecules or molecules that have a specific metric or property? We use the metric QED, which stands for a quantitative estimate of drug likeness. It uses a couple different parameters and is one of the ones used sort of throughout research as a way of showing an improvement, especially in terms of properly pruning that search space towards particular candidates. The larger problem is it's really expensive to test these drug molecules. Some of these, it'll take a year or two just to be able to validate something, plus any trials that are needed for that. So the goal is to really be able to use that time where there is some sort of wet work in a lab that needs to be done, making sure that it is done in a way that's well directed, because it is an expensive process. Further, are there things that we can discover that we may not be able to otherwise? So we'll first start by taking a look at what SMILES is. I think this is an important thing to understand and applies to really all of our methods. Is the base of all of our data are SMILES strings from the Zinc database. SMILES is sort of exactly what it looks like. It's the simplified molecular input line entry system. It follows a particular grammar in terms of how cycles are made, how we're able to show bonds that are extending off the benzene ring on the left, etc. But just a long string to which we represent those. We used a subset of the Zinc data set for our project where we were looking at particular portions that met some characteristics. So we'll start with matched molecular pair analysis. So this is probably the oldest and the sort of the baseline technique that we looked at to compare all of our other methods against. The idea with MMPA is to do a rules-based transformation. So what do I mean by that? Well, if we look at the aromatic two molecules on the bottom left, we see that they differ ever so slightly. There's the hydroxyl groups off the benzene ring on the left, and then there is that oxygen that's inserted on top of that. So how do those differ? Do those have different chemical properties? Perhaps one has a slightly different solubility. Perhaps one tends to be a bit more drug-like in some way, or some other sort of metric. So what has been built up over years and years of a massive database of these matched molecular pairs, where we're able to say that a particular transformation tends to mean this thing in terms of its effect. So matched molecular pairs are, at a very aggregate level, the idea of aggregating a large number of those pairs and then applying a set of transformations with the goal of making that effect. So we can see sort of in the upper left breaking down one of those fragments. So we get a fragment, we index it, we load up what we know about it, like its solubility and such. We then match a bunch of the pairs, apply some transformation rules, and then predict the effect. And then based on that predicted effect, we can then make some more informed decisions about the molecules that we can look at. MMPA, been around for a while, sort of a bread and butter of cheminformatics, sort of in mid-'80s. Now there are deep learning methods which are actually much more effective in many of these studies. So one that we'll look at is, and one that we looked at, was grammar variational autoencoders. This sort of takes the idea of, you can imagine applying just a variational autoencoder naively. That's actually a particular method called character variational autoencoders, and that's where you're encoding from the smile string directly to another smile string and then rating it based off of that. So you're using really like the smile string as your initial molecular representation. The trick with grammar variational autoencoders is to say, that string maybe not the best thing to represent it as. We run into problems of having invalid states. If you just have that string and you output another string, that string might not actually be valid. So grammar variational autoencoders, we can see this in the diagram on the right-hand side. It essentially forms a parse tree, and then it uses that parse tree to figure out the rules, encodes those rules into some vectors, and then maps that into a latent space, and then does the variational autoencoder thing to then generate those smile strings. This method is incredibly effective. It avoids the invalid state, so we're not necessarily exploring a space that we don't need to explore, which is just inherently more effective. We're not sort of wasting the cycles on that. Junction tree variational autoencoders is then an extension of that. There's the idea of, okay, we can use variational autoencoders, but our method of representation junction tree variational autoencoders extends that. So we know, okay, we could use these smile strings and parse them and then use that parse tree to get some rules. But what if instead we use a different representation, a representation that might more closely match what we're trying to do? So junction tree variational autoencoder says, okay, we'll do two things. We will, one, represent it as a graph and then directly encode that graph into a latent space and use that. But we will also do something else. We will do this process of tree decomposition, which is a specific algorithm which essentially chops up that molecular graph into its substituent clusters and then encodes those clusters. And those clusters are sort of inherently more representative of the scaffold of that particular molecule. They are then essentially, it uses a process of message passing, and there's learning is a little bit more complicated for this one. It does use some Bayesian optimization, et cetera. But then it decodes that into a final state. And this has a sort of state-of-the-art performance. We had some troubles with this method because it required a fair amount of training time. We were able to get about 35 hours or so of training, and we got to like just above baseline, needing to likely make some hyperparameter changes. But we got results that were better. And then finally, there's graph convolutional policy networks. So this takes really a different approach from the others. It doesn't say, what is the way that I can apply a variational autoencoder to represent the latent space in a more effective way? Instead, it says, okay, I have all these graphs. Um, how could I go about approaching this? And it does this by building up these scaffolds and then defining a set of actions that it can use to then generate potential states. And then it rewards those based on how well they fulfill a specific set of criteria. In this particular example, we call out, does it meet the valency checks? If it doesn't, okay, that's not going to be a good one. So don't reward that. Don't do adversarial learning for that. And then put those together. So we went through those methods. We did have a fair amount of trouble recreating some of them sort of to the same state-of-the-art results that we saw in the papers that we were looking at. We did sort of get the general takeaway that while there are a lot of methods, one of the big ones is the representation seems to lead how effective we are and how we're able to apply some of the methods. This really goes back to the initial slide, which I am graciously stealing from Professor Sun's lab, that learning a molecular representation is really how we can sort of represent and approach a lot of these problems. So I think that was an effective and interesting thing for us to learn in this project. That's it. I would like to thank Professor Sun and the TA team for helping us throughout the class. I think we got some interesting results. This was a fun project. It was something that I didn't think I would ever explore. So really sort of gave us a unique approach on that. All right. Thank you very much.